Thank you. It's good to be here. I apologize. I'm fighting an illness right now, and I should be a good virologist and cover up completely. And I also apologize because I had to compress a talk that I recently did in order to give you something to hear about today. Typically, when you talk about March Madness, everybody used to think about basketball, but today it could mean politics or any other things going on. But I wanted to share with you a journey that I experienced some number of years ago when Ken Burns was recruited from Johns Hopkins to the University of Florida as the youngest chair ever. And he brought along with him his postdoc, which was Bill Holloman. Bill Holloman was instrumental in doing the first AAV gene therapy in the eye. Right down the hall from Ken was Dan Nathans, who just got the Nobel Prize for recombinant DNA technology. And Ken pulled out of his lab a postdoc named Nick Muzichka, and then he went to David Baltimore's lab and he pulled out two young superstars. One was Bert Flanagan. I couldn't get the picture of Ed Braun. And the last of this Fab Five was coming out of Charles Radding's lab at Yale, Bill Holloman, who just came from Holliday's lab where they figured out the Holiday structure. And so it was a time where I was fortunate enough to be one of the early students to come to a place where the young superstars coming from all the labs that were doing this type of work. I'll point to you, this is what we call deep sequencing back there. In the old days, used to put multiple films against the plates to get your read. They were sequencing AAV. This was a thesis project, took 4.5 years to sequence 4,681 nucleotides. I share this with you because about 2 hours away from where we were doing our work, they were launching the Voyager spaceships. And so it was always a curiosity of everybody in the lab is, where are we going next? If your guys are too young to remember that. During this time, when Kennedy was president, said we were going to the moon, it was a president you could believe and stuff like that. And so we were all enamored that the sky was the limit. And this is the first photograph ever looking back at the moon and the Earth. And so we were convinced we could do anything. And this is the kind of atmosphere that was taken on at that time at the University of Florida. You have to appreciate the fact that everybody knew what a biologist was back then. People are also familiar with microbiologists. We were trying to introduce a new term called a molecular biologist. And we clearly didn't rank in this order, but it was a new concept, believe it or not. We had to build a P4 facility in order to grow PBR 322 bacteria to do a recombinant DNA transfection experiment. What was ironic, the carboil that we took all the wastewater out, we splashed all the way down the hall to the autoclave. It was all done in P4. For that reason, anyone who's not familiar with AAV, and I see too many of my former colleagues, L'Oreal, Don, Doug, David, so forth and so on, that I'm actually embarrassed to show this, but this is an illustration of Jenner, who was vaccinating his kid with cowpox. He noticed that the women that were milking the cows in London never got smallpox. They had pox on their hands, so they were getting a version of cowpox. And he reasoned that if he could take that infectious agent and put it in someone, he might protect them from smallpox. So he actually, without IRB consent, took his own son and inoculated him with cowpox and then gave him smallpox. So actually, this was like one of the first examples of gene therapy where you're genetically educating the immune system to redevelop itself so it could fight a disease. So I had the embarrassing experience of going to China and showing this result, thinking I was educating And after it was over, as polite as they are, one of them tapped me on the shoulder and said, and the Chinese were doing this in the 14th century. And I was like, oh my God. So you have to envision that without understanding genetics or anything involved, they were able to carry out what were called simple concepts, genetic material from the outside getting into the inside of a cell. And then after that, something would happen that would either be positive. So I'm going to take you down in our world. This is the head of a pin. If you go up in magnification, that's a human hair. And unfortunately, I had experience at some of our gene therapy meetings. This is a bed mite. As you go up in magnification, there's a yeast organism, red blood cell. And at 2 microns, on the left is E. coli, and on the right is Staphylococcus. And at 200 nanometers, that funny-looking thing is actually Ebola virus. It has a cane-like shape. And if you go down to 20 nanometers, this is an AAV particle. And technically what we're talking about is a non-envelope icosahedral capsid that carries a single-stranded DNA genome of about 6,000 base pairs inside of the nucleus of this capsid. So I'm going to try to cover roughly 60 years in 30 minutes, and I'm hoping that I don't insult or anger anyone that I might leave out as I try to cover this information. But this journey, it was fortunate that I was able to participate in it from an early start. But in order to give you a historical perspective, let me just pull back the curtains and take you back to the 40s and the 50s. And if you're old enough to remember this, I was born in 1954, so my older brothers actually went through the polio epidemics. This was a time where it would run across the country and thousands of people, mostly kids, were kept alive in what was called the iron lung. And these pandemics were every season, to the point that, Time magazine, it's this the year that we may get a vaccine to actually protect them. But there was the same kind of hysteria back then that we experienced with COVID. The kids were fleeing the cities. In certain towns, you couldn't bring children into those towns in order to keep polio out. You were barred from going to various places. And there were such superstitions of what was causing polio. They actually killed something like 8,000 dogs and 3,000 cats and stuff, just trying to find a source that was responsible and get rid of it. So again, without understanding the etiological agent that was involved, the hysteria of how do you fight diseases like polio was being mounted by a research community that itself didn't know what it looked like, but were able to eventually make a vaccine. That vaccine, when I was young enough, was called Saving on Sunday. You stood in line, hundreds of people, and you walked out, they gave you a sugar cube, and they put in attenuated polio virus, and you swallowed it. If you know anything about polio virus, it replicates in the epithelial gut cells, then it migrates to Pryor's patches, and after that it gets into the nucleus or the horns, and when it replicates there, it kills the neurons, basically paralyzing those muscles. So while it had a tropism for neurons, it actually had to infect through the gut. So the attenuated virus would replicate in the gut, and your immune system would mount an attack to prevent it from going all the way to polio. We did a fantastic job of getting rid of polio in this country. In fact, the only incidents of polio were actually from out Midwest, where farmers who had grandchildren that had the polio vaccine, and when these grandparents were changing the diapers, they were getting exposed to polio for the first time and coming down with severe disease. Right now, this is technically eradicated off the planet, except it requires 4 degrees in order to make this vaccine. So the third world countries, when there's an outbreak, there's what's called a surrounding of it and vaccinating so it doesn't go any further. I don't need to tell you what's happening now with measles and other things that we can expect these kind of flare-ups to be more prominent. But because of this, there was an effort to go find a virus that was causing the common cold. And the idea was is if we could make a vaccine to that, we could stop all of these work days that were being lost for people being sick like me right now. And in 1953, a group at the NIH took out of the adenoids of patients that had upper respiratory infections a virus they called adenovirus. And this virus, which had been characterized and studied for a long term, they noticed that in the middle, when they were looking at it, they would see periodically these small structures. When they purified them and put them on cells, they did nothing. So they thought it was a precursor protein that eventually turned into an adenovirus. And it was left dormant for 10 years until a group of individuals came along and started characterizing this. And this was done by Atkinson and others, where the name adeno-associated virus was stuck because every time they grew this virus, they kept seeing this little virus come along with it. And so the curiosity was, what is the purpose of this virus? And why does it have to grow with a helper virus in order to propagate itself? This has never been seen before. You would have never found AAV if you were looking for symptoms, because they did an experiment that you can't do today, once again. They went into orphanages and they gave them adenovirus and adenovirus plus AAV to see if he had a worse symptom of the upper respiratory. And they were not able to show any clinical symptoms, so they called it a non-pathogenic virus. So it's almost like a molecular symbiotic relationship that exists between the virus and the host. And this was enamoring to us at that time because we were looking for a delivery system that you didn't have to uncouple the pathogenicity, herpes virus, adenovirus, and so forth. So you could see in the early 60s the number of papers that trickled out, and then basically how this whole field has exploded. And this is primarily responsible for three people that I was fortunate enough to work with. Mavis Evganji McKenna, who solved all the crystal structures for AAV and basically gave us a GPS roadmap. Sorry. Nick Mazicka, who was my mentor and a fantastic individual, that was basically, let's go, let's do it, there are no bars. And of course, Ken Burns, who was a genius behind all of this, that put this Fab Five group together with a bunch of young graduate students. So the concept's simple again. It's like Jenner. We were able to dissect the life cycle. That is, if you put AAV and adenovirus into a cell, they bind to their respective receptors, they go to the nucleus, they uncoat, and they carry out all steps of macromolecular synthesis in the nucleus, and then they release their progeny. But if you take purified AAV and put it in the cell, and there's no helper, it goes latent. And in the laboratory, this was evidence that it was jumping into a chromosome. This latency is basically genetic suicide, except if you take that cell and infect it with adenovirus, it'll jump out of the chromosome and amplify. So it's like the perfect hitchhiker. When there's a carrier to take it to the next cell, it goes. When there's no one there, it goes dormant. And if it gets infected, it amplifies and goes again. So we reasoned that if we could master this portion of the life cycle, we could turn that portion into molecular FedEx trucks. And this is basically the project that I had in Nick's lab, starting in 1978 at the University of Florida. Again, you guys are probably too young to realize that this was the Bible back then. Tom Maniatis' cloning book, everybody used it to learn how to do cloning. And it was in Nick's lab that we actually cloned the AAV genome into a plasmid, PBR322. Again, you guys are too young to remember this. PBR322 was Roy Curtis's name for Pabst Blue Ribbon, 3.22% alcohol. Everybody had some clever name at that point in time. But what we realized was that when we cloned this, we could make milligram amounts of this DNA and put it physically in cells, and it did nothing. But once you added adenovirus, it would jump out of the plasmid the way it rescued out of the chromosome, and it would amplify itself and make virus. So this became the tool that one could start with a single unified molecular substrate and start testing and being able to get it to go through its life cycle. In doing this, we proved a very important hypothesis that Ken Burns, which was that the AAV terminal repeats go through a self-priming replicating mechanism that Doug McCarty and others validated at the enzymatic level, and that it would go in and flop because it was a clone, but it came out flip-flop, which meant it was replicating back and forth. This was the same substrate that Nick and Paul Hermanet showed the first gene to be cloned and actually transferred into a cell. So this was novel. This was being done with retroviruses. This was being done with adenovirus. This was being done with lentiviruses. I was fortunate enough to go to Princeton, where Arnie Levine was building a new molecular biology department. It wasn't just a molecular biology, it was a department. And I was able to work for a young scientist called Tom Schenck, who happened to come out of the other half of the Nobel Prize for recombinant DNA, which was Paul Berg at Stanford. So Dan Nathans and Paul Berg shared that prize, and I felt privileged that I was being trained by both of these young individuals. Gave me insight. In Tom's lab, we went through the traditional virology of separating what's called cis elements from trans elements. And basically what we were able to do was to put the AAV terminal repeats on a molecule separate from the AAV coding sequences. And if you threw those two plasmids in a cell and added adenovirus would turn on the rep genes, they would rescue the terminal repeats and replicate it, and the capsid genes would start packaging this DNA. So it was like one of the first packaging systems that came along, but you had an adenovirus contaminant. And back then, this was fairly straightforward because adenovirus was heat labile. We just put it in the water bath for 45 degrees for 30 minutes, killed all the ad, and then used the AAV to go forward. We eventually, from the work that Tom did, we deciphered the adenovirus genes that were responsible for making AAV grow. And we put those on a mini plasmid and used that to put in 3 plasmid DNAs, but only one virus came out at the end, and that was the AAV carrying the gene of interest. So this is, again, on the shoulders of others building a walkway that began to give us a runway for how do we develop drugs and going forward. Steve is here today, and Josh were the primary ones that optimized this process. And what we were able to show is that we could take it from small shaker flasks to now 2,000 liter bioreactors. And this is being done commonly and routinely throughout the industry, but you heard a little bit earlier, the price of this needs to come down and the efficiency by which it's made needs to go up. But I'll speak for 1 minute at the end about that. I don't want to mislead anybody. There have been multiple ways to make AAV by many talented individuals. I'm just describing the triple transfection that we ended up optimizing and how it helped facilitate the field going forward. Between 86 and 93, I went to Pittsburgh, and I was working on trying to find AAV latency. We discovered it jumped in chromosome 19, and we started some collaborations with the guys at the NIH looking at sickle cell and hemophilia and so forth. And I got recruited by Bill Marslet down to North Carolina, and through the Biotech Center, which gave us one of the first grants ever to build a gene therapy vector core facility, we actually put in place a facility that could produce virus, not just for us, but for the entire community. And that facility, eventually run by David and others, made virus that went around the world. A very impressive group. But there were 2 studies that came out that separated the AAV from the other viral vectors. One was done by Mike Caplet and During and Zhao Zhao, where they took AAV with a reporter gene and stuck it in the brain of a mouse and showed that 4 months later, it was still on. And that was exciting because 1, it went into the brain. AAV is an upper respiratory virus. And 2, it was still expressed after 4 months. And so a lot of criticism was, oh, the brain's immunoprivileged, it's not going to work if you put it somewhere else. But Zhao Zhao went and did an experiment where he took mice, normal mice, and he put a LAC-Z gene in it and showed that over a year and a half later, these animals were still expressing the protein. And this was unheard of because if you use naked DNA, retroviruses, or herpes, as soon as you put that gene in there and expressed the protein, the immune system came and cleared it out. And so we thought there was something unique about AAV's latency, that it didn't alert the immune system to attack the cells. And I think this study, along with others that were following behind it, kind of launched 1,000 labs to start looking at AAV as a vector for, and which is why all the publications go up, like we said. You should realize that Rob Cotton at the NIH, after we worked on AAV2, he cloned AAV5 and showed that it could work. Wei Dong, who was a graduate student in my lab, went to Jen's lab, worked on AAV1, and you could see these were the early viruses that became drugs because they were the first ones to be characterized and turned into production systems and allowed to go forward. So I'm going down this timeline, and at the same time, I'm trying to bring you hallmark experiences that the entire field was exposed to. And this was a study carried out by Avogen, where we were convinced this was going to work for the low-hanging fruit of hemophilia. And what they did was they took a subject with hemophilia, they put in AAV with the factor IX gene, and they got expression, and it lasted for a month. But then liver enzymes came up, and the expression went away, and it was gone. And nobody knew what was going on because we did mice, we did dogs, and we did monkeys, and none of them had this phenotype. But in this setting, in a human, the immune system said it was attacking this therapeutic cassette. And they dropped the dose tenfold, and they no longer had the immune response, but they didn't have enough therapeutic gene expression to make it meaningful. So all of a sudden there was a ceiling, and there was AAV biology. How do we stay under this ceiling of toxicity but bring biology to get us a therapeutic? And this work was done by a brilliant colleague of mine, Doug McCarty, who actually went in and said, the rate limiting step for AAV is going from a single strand to a double strand. So if we can make a double-stranded molecule and package it, as soon as that virus uncoats, that DNA comes out, transcription starts immediately. Whereas in this setting, you have to wait for a round of replication or re-annealing to take place. And when Doug validated this technology, it immediately went into the clinic. It's actually the therapeutic product that SMA, Zolgensma, is used today. So you can see, as we were understanding the biology, therapeutics were popping out left and right as far as capsids and replication and technology. There was another inflection point that took place from my perspective, and that was when Joe Rabinowitz in the lab, working with Don and others, came in and showed that they could cross-package. Joe wanted this title. Believe me, we fought with the editors for a long time. That he could take the same genome and package it into any serotype by building a chimeric packaging system with the same rep proteins and different capsids. And Guangping Gao and Jen's lab had just uncovered all these clades and new AAV vectors. So what did that mean from a research perspective? A graduate student or a summer rotation student could take an AAV plasmid, delete out the sequences in the middle, put in the gene of interest, and then simply package that into any serotype you wanted. And this is the playing field that the community has today, based primarily on these guys and this engineering of these production systems. I'm compressing the next 20 years because it's probably been all contributed by you guys, and I don't want to embarrass myself or insult anybody. But you can see there's been a tremendous amount of work looking at how do we optimize the AAV capsids, how do we get them to be more efficient, how do we get them to be more effective, and at the same time produce them quicker, cheaper, and so forth. What I'm going to do is toggle back over to where we were from the world that was understanding AAV biology and the world that was trying to rescue patients that were dying of genetic diseases. And the first example, which was really a Herculean effort, was done by Terry Flock, who in 1996 took the simple logic of CF is an upper airway respiratory disease, AAV is an upper airway virus. Let's put the CF gene into AAV and put it in the lungs and we'll cure them of cystic fibrosis. They carried out this trial, made virus, got it purified and everything, and they went into the patients, but they didn't see any results. There was basically no evidence of gene transfer. This was puzzling to us because everything we knew about AAV kind of collapsed. And why didn't it work? And what we ended up finding out was that Candace in the lab discovered the first receptor for AAV2, which was heparan sulfate proteoglycan. Co-receptor was an integrin. And Jeff Bartlett started to show the infectious pathway. So we actually started to figure out how AAV got into the cell. And on airway epithelial cells, there's no heparan sulfate. It's only between the membranes. So you actually have to create damage to get access, which is why adenovirus, which technically creates a pathology, allows AAV to get access to lung cells. So it didn't make sense to go in and damage a lung that's already sick to get a therapeutic in. So we and others started to look at other targets that AAV may be more useful. This image was done by Arun. I stole it a number of years ago because we're best friends. He now steals my image and uses this, but this is to show you that over these years, we've gotten better at understanding the steps that are required to get this virus from the primary binding to the receptor to endocytosis, trafficking, uncoding, getting into the nucleus, and so forth and so on. And it's still a quagmire because we can't say what happens in the lung may not be the same as what happens in the brain as far as uncoding and stuff like that. But all I want to do is let you know that with that low level of comprehension of biology, we were starting to go into areas that we'd never been there before. And I think the Voyager is an appropriate thing to bring up now because at this time, Voyager was leaving the heliosphere of our world for the first time. And they gave it instructions to turn the camera around and take a picture of Earth. And that's that little dot back there. And so you can imagine where we were at compared to where Voyager was at and doing something that had never been done before and seen before. And we carried out what were the first gene therapy experiments for Duchenne muscular dystrophy. I apologize, I'm getting a little winded. There was a meeting this week where in Dallas, Texas, where they were describing these very early studies. And these were crazy experiments. We didn't know what the hell we were doing. We made a chimeric virus that Don made in the lab that was called AAV 2.5. It worked better than AAV 1. 2 was the only virus that the FDA approved, so we asked them if we could put this in kids, and we injected these kids with this virus and showed that we got a gene expressed. At the same time, today you probably heard there was a death in the Sarepta trial using this gene therapy approach, which screams at us that we are not where we need to be yet, and I'll talk about that a little bit later too. But I just want to leave upon you that the field exploded while we were understanding the biology. The drugs started popping out left and right for, thank you. I'm so sorry. I'll keep pushing the buttons so you can see what's happening. And as this was going on, all of these drugs were getting approved one by one. I think one of the most remarkable things is that every drug that will ever be approved by AAV will have the 145 base pair inverted terminal repeats that Ken Burns characterized, and we cloned in Nick's lab some 35, 40 something years ago. So, it's real easy to show these things as, they're bottled, they're drugged, they're out there, but I don't think until you see how it impacted people's lives, you can appreciate what you guys are actually pioneering out there. And these were some of the early studies that were done.
To measure the patient's performance before and after the treatment, he was guided through an obstacle course. Here, you can see how he navigated through the maze prior to the surgery. He bumped into obstacles 6 times and was disoriented twice. It took him 77 seconds to finish the course. 6 months after the treatment, the patient showed a dramatic improvement. He was able to make his way through the maze in 14 seconds without bumping into anything or getting disoriented.
So this was our first drug, Luxturna, with AAV2. Bill Houseworth, that I showed that picture, was responsible with Gene Bennett and UPenn in getting this going, and then eventually Gene and Spark took it all the way through. This is Doug's self-complementary drug in an AAV9 capsid from Romping Gal, treating these kids that had spinal muscular atrophy. And this was one of the first patients we did with systemic gene therapy here at Duke, actually. This was Connor playing with his video game while he was getting ready to be dosed with 10 to the 14 viral genomes per kilo. And the mandate on when Doug ran the facility and David ran the facility and so forth and so on was, you don't do this unless you're willing to put it in your own kid, because of the potential outcome that could go wrong. And Connor, when they were doing this, we were outside. Eddie Smith, who's my neighbor, came out and said, "Jude, he wants pickles. He's eating pickles like crazy." I said, "Eddie, forget it. He's a sour nut. It's got nothing to do with AAV." And then I ran back and I was looking at the virology book. AAV goes through the gut. Maybe it's acidic. Maybe he's trying to, I don't know. We all went crazy that day. And to get into trial, you had to do 5 steps.
Great job.
And then this was him about a month later. This was Owen, the second patient, and you could see pre and post. And I show these images because we made this virus and we took it there, and Connor wants to run up the steps in Philadelphia, and Owen wants to be a baseball player. And Sheila and I were able to take him to the Durham Bulls game, and you could see that these boys were thriving. And Connor has come back. He's 7 years out from his first, and he's still doing well. But this trial failed for us with Pfizer. When they went into a large-scale study and carried it out, they did not get enough difference in their markers to show that it was therapeutic. We were, of course, frustrated because actually the FDA said you really need to be careful how you do gene therapy experiments. You can't do placebos and controls because you lose all the things that happen that are in between. That's another story for another day. But we are running into a problem that we could treat a kid at this age, but as he grows up, how do we do repeat administration? And so I don't want to leave you with the impression that this is a Wizard of Oz, the stories of the Yellow Brick Road. We have lots of things that are confronting us today. Some of them are being approached and others aren't. Large-scale production, I'm convinced that what Josh and Steve and others did, going from shaker flasks to these large bioreactors, is an industry standard. But we can convert this to an in vitro replication system where you just do it as an enzymatic reaction. And I think Doug and Nick showed some of the first papers to illustrate that. And I took this cartoon from Lonzo, who was showing how you can make T cells for individual patients. I think you'll be able to do AAV production in an in vitro reaction where you replicate the genome like we do with doggy bone and then package it into a capsid and just purify it from that step without going through the traditional infectious cycle. The transduction efficiency, we've all lived through this. We've gone into rodents, monkeys. We're now doing organs on a chip. Don's lab was the first to kind of look at a organ where the heart, where you pull it out of a target, transduce it, put it in an animal, keep it alive. This was an effort that we were doing. This is a lung that was out of a CF patient after he got his transplant. We were using these lungs as they were breathing to send AAV libraries through it to see if we could find new vectors that would target the lung. So you can begin to see that we're going full circle. We're going back to the future where what we started with rodents and found out that that's not the same as a human. We now are in humans trying to find these delivery systems and these zip codes that will get us to the right target cells so they can be therapeutic. I'm going to end this by letting you realize that we are not there yet. In my mind, this was a travesty because it should never have happened. He had liver failure. We have capsids that detarget the liver that still go to muscle. The problem is you have to get in line to take it all the way through these clinical trials instead of swapping it out. At Aspire, we carried out one of the first studies that I thought was important. It was empty particles put into healthy volunteers to see what really happens when you give them the virus, not the transgene. And we went through and we did a number of patients and we measured their T cells and so forth and so on. But the observation that we got that was interesting was this diversity of response. And this is what we don't see when we go into our animal models. We don't see this diversity. And so what I'm trying to convince you is that the field needs to start moving in a direction that's available to us, and what I call it's called the collaborative cross, where they take the 9 founder animals that we all use, most of us use black C57, and they have bred them into different generations of offsprings, and then they have inbred them so they lock that in. So every gene is known which parent is donating it in these lines, and they completely sequence. So if you get an observation in one of these mice, you can trace it all the way back to which gene is responsible for that observation. I'm going to show you the data that we generated taking clinical material and putting it in the classic C57 mouse, putting it in the non-responders, but now finally putting it into the collaborative cross. And you can see that diversity opens up. So we're at a crossroad here where we can finally start mapping in models who's going to be a responsive person to an AAV therapy and who's not. And I think we don't have a choice but to go down this arduous path of trying to find out why is mouse strains low and why is such a strain high, what genes are responsible for that. We do it today when we look for neutralizing antibody. There's no difference to look for other factors that may determine the outcome. In doing that, I'm closing by telling you it was a long journey in which we spent an enormous amount of time trying to get from a basic science and quickly passing it off to our clinical colleagues. This is probably not the optimum way to do it. It would have been great to have a 20-year effort to learn the biology and then bring that to the community. But we went from virologists to FDA production people to IND people to IRB, and the community here really deserves a big hand. This came out of my thesis. With no cure for the 2,000 human genetic diseases, viral vectors in the future may transfer normal genes. The most important thing about this statement, shit, it's going from 2,000 to 7,000. We're losing this race. I'm just going to finish with this following. We're on this path. There's now like 290 clinical trials with AAV that are ongoing. So I don't think we're anywhere near slowing down, but we're just revving up. And I'll remind you, it took 4 billion years to get here. 3,000 years to build these triangles, 2,000 years to build the wall, 600 years from the first rocket to go to the moon, 60 years to go from the polio vaccine to maybe living to be 140 years old. It's scary. From wrinkled peas to Watson and Crick. Look at this, guys. SMA was described in 1890. They described the disease in 1890. Almost 50 years later, the sequence, you had vectors, you had Doug's cell complementary, you have SMA mouse models, monkeys, you have the first drugs, and then it's approved. The reason I show you this, I was teaching a class in Spain when a newsletter came out and it said, an individual was born with SMA, and the doctors at that point in time didn't know what to do, was in critical care. So after the first 3 days, they said to take the child home and just love them. But one of the researchers said, wait a minute, there's a drug. And they stopped them before they got in the car and left and brought him back. And within 4 days, he got his life-saving therapeutic. So I leave you with the idea that time is compressing very fast in this field, and most of you guys are going to be responsible for making it even go faster. And I'll stop there because I can't talk anymore.
We have time for 1 or 2 questions, if I have questions in the audience.
If you're someone from my lab, I'll shoot you.
Hi, thank you for the wonderful presentation. It's always very inspiring. I was intrigued when you said that you think that AAV might be going in vitro, like doggy bone DNA.
Yes.
And I was wondering how you envision that. Do you envision also the packaging of... So this is something I've been thinking about for the past 5 years. And I did find
young. Go ahead.
I feel like there are a couple of challenges. I did find a paper where they were able to package AAV genomes into capsids, but they used nuclear extract.
Yeah.
Yeah.
That was Doug and Nick. He's back there. You can harass him.
Thanks for the favor. But then I was thinking, one, we don't know what factors are in that nuclear extract.
Let me help you out and get you to the punch. So if you take the 5X polymerase that does single strand, you spin off single strands like this, and then you throw in the AAV rep protein, which will cut it at that. Then you just need the AAV capsids in there at the same time to do a packaging. A paper just came out from Mavis's lab showing that packaging looks like it may happen as a condensation step where AAVs thread through the 5-pore entry. So there's the potential to probably put in an energy like ATP in the capsid while it's replicating and spinning out. The rep protein more than likely is going to dock to the 5-pore and clip it every time it goes in. So you'll package it like a biochemical reaction. I think Doug can give you the details.
And then my follow-up question is about DNA methylation and about post-translational modifications of the capsid proteins. So this could be something that is beyond the capability of an in vitro system, or do you envision also adding DNA methyl transfer?
Can I get your name? Could you? You're asking a very relevant question. So I have a beef with the FDA because they tell me we have to purify these to homogeneity. And I keep telling them AAVs never purify the homogeneity. When adenovirus lyses a cell, it's through passive lysis. It attenuates a P53 mechanism and the cell lyses and AAV spills out. That means it's constantly associated with other proteins. And yet we're purifying it and stripping it away to put it back into a cell where it immediately gets glomped on with protein. And there's a number of papers out there that show this affinity. So you are absolutely right. When we take naked AAV particles and put them into a patient, we're basically rolling the dice which proteins are going to assemble around that capsid to carry it to the next step. I think we should be doing the post-modification translation to those particles before we introduce them, and realize that can happen in the production system, that can happen during trafficking, and it can happen in the nucleus. So there's a lot of biology that we're just blowing up when we do this purified in an FDA bottle to put into a patient. So I think you're really right on to something. In my opinion, the in vitro systems will at least give you a cheap way of getting lots of material. And then after that, you can work on the modifications to get higher transduction. We have a paper coming out in the 25th anniversary issue of molecular therapy that I think is going to address some, if not all, of these concerns. So I steer you that way.
Thank you.
Great. Thank you so much.